Well, good afternoon. My name is Ala McCoy. I am Director of Startup Support at the University of Maryland's UM Ventures. And I would like to extend a warm welcome to all of you who joined us today for the next installment in Startup Fundamentals workshop series, both live webinar audience and those watching a recorded version on our Startup UMD YouTube channel. This workshop series is offered by UM Ventures with collaboration from Maryland Technology Enterprise Institute at the Clark School of Engineering and Dingman Center for Entrepreneurship at the Smith School of Business. To everyone attending the live session, please feel free to submit questions using the Q&A function. You can usually find that on your screen. We are recording this session. It will be posted on our Startup UMD YouTube channel. So if you wish, you can submit your question anonymously. Just select that option. So today I'm very happy to welcome Dr. Murat Kalayoglu. He is a co-founder of Cartesian Therapeutics. He has served as the company's president and CEO prior to taking it public through a reverse merger in November 2023. Murat founded also two other successful biotech companies, which is extremely rare. And so we're very happy that you are here today to share your wisdom with us and your experience. What it takes to build a life sciences company and unique characteristics and challenges of this industry. So the microphone is all yours. I will be behind the scenes monitoring the Q&A. So if there's anything, just let me know, but otherwise the mic is yours.
Okay, I appreciate it. And thank you again for setting this up. In preparation for today, I was reviewing some of the previous sessions that you've recorded, and I think there's a lot of great information, I think, for aspiring bio entrepreneurs through the YouTube channel with these pre-recordings, as well as all the resources that you and others at UM Ventures have put together. So I appreciate that, and I'll reference some of those links to for the benefit of the audience as we move forward. So I have a few slides that I'm going to see if I can pull up here. Let's see. I'm going to do this, make it full screen, and then share. Let's see. Here it is. Okay. So if anyone can't see this, this title slide, Building a Biotech Startup, please let Ala know, or somehow, let me know through raising your hands or whatever you need to do. But I'm just going to assume you can see everything here. And I'm also going to assume that the audience for today is going to be University of Maryland Life Sciences researchers, whether you're faculty or on staff and any other capacity, whether a graduate student, et cetera. I'm going to assume that you have some interest in starting and potentially building a startup to commercialize your venture. And in particular, we're going to focus on biotech for purposes of today. We'll give some examples from a healthcare IT company that I was involved with to make a few points along the way, but the majority of the talk will be focused on starting and building a biotechnology company. So we'll talk about it in different steps. So we'll first discuss discovery to company formation. Then I'll give you a sense of what preclinical development might look like from the eyes of the entrepreneur, then clinical development and early, mid, and late stage clinical studies. And then we'll shift gears and cover a few topics that I think that I hear a lot of questions around. Different approaches to biotechnology operations. So virtual company versus a brick and mortar company. What do you do with manufacturing? Financing, different vehicles that are available, conventional versus alternative sources of financing. And then finally, liquidity exit, whether it's an M&A or taking a company public. We'll have ample time for Q&A. I encourage everyone to chime in and ask questions along the way. Alla, maybe you can just jump in and ask the questions that as they come up. I'm happy to take the conversation any way that the audience wants. It's really a benefit for it's a it's your time that, and we want to make sure that you're learning as much as possible. So, and again, along the way, I'll draw in examples from my personal experience through a handful of companies that I've been involved with. Just very briefly, Health Honors, the name Med Incentive was an older name. I put that in because it was the name on the first slide deck we ever put together. So you'll hear that as well, but it's the same company. Health Honors was a way to motivate people to take their medications on time and as scheduled. Topokine was a small molecule that we took from concept to late stage clinical studies before selling it to a pharmaceutical company. And it was a medical aesthetic that you, in essence, applied to underneath your eyelid to reduce fat. And then Cartesian is a cell and gene therapy company that is more of a brick and mortar operation that we started in 2016 and have and are currently focusing on a CAR T approach for autoimmune diseases. So 3 different companies, 3 different experiences, and I'll again just draw on some of them to make some larger points along the way of us covering these topics. Okay, so the first step here is, again, we're going to assume you're a life sciences researcher, and you, in essence, come up with what you believe is a new invention, sort of an aha moment, and you are interested in commercializing that discovery through company formation. Before you do anything, I would recommend that you clearly articulate the problem, the solution, the solution's unfair advantage in solving the problem. In essence, the problem is, what is the problem you're trying to solve, and the impact of solving it expressed both in terms of the patient as well as in terms of economic terms. Number 2, once you've articulated the problem, what is the solution to the problem? And describe that in a level of detail that's appropriate for the audience. So it's not necessarily, if you're about to present your discoveries in an academic conference, your solution to the problem or the indication is not going to be appropriate for communicating it to an investor audience or a tech transfer office, et cetera. And then the third is, what is the unfair advantage associated with the solution that you've developed? You can think of that in several different ways. How similar is your solution to other solutions, or why can't other solutions be just as effective or more effective? Or how can the solution be protected, whether it's through patents or trade secrets? And finally, once you've come up with that, with that equation, then you can ask, what other problems can the solution be used to solve? What would be the impact of doing so? So I want to go through a relatively straightforward example to sort of highlight these points through a company that I helped start. This is the very first slide deck that we put together, and I just grabbed the slides that reinforce this algorithm of articulating the problem, articulating a solution. What is the unfair advantage of that solution? Again, it's not specifically biotech, but I think it gets the point across. So when we articulated the problem, we did it both in terms of the patient, but as well as in the economic terms. So this is, I think, slide 2 from the deck. Non-adherence to medications, non-compliance to medications is a big problem because drugs don't work in people that don't take them. And so when patient compliance is only 50% for chronic diseases, then that creates a real issue in treating that disease. But it also, from an economic perspective, really creates a problem for our eventual customers for this technology, which are the pharmaceutical companies, such that we calculated that enhancing compliance adherence for a single blockbuster drug, and at the time, Lipitor, by even a third would boost Pfizer's revenue by $4 billion. So we wanted to articulate this in very clear terms, and then describe the solution to non-compliance. So we came up with this technology that could motivate patients to take their medications on time. And in the process, we could boost adherence compliance by a third. And our solution and our sort of was, in essence, paying people to take their medications on time, but paying them not through the typical way, you do X, you get Y, but rather through Skinnerian conditioning, intermittent reinforcement. So sometimes if you took your medication, you could log in or call an automated line and tell us you took your medication. And if you did, you had a possibility, but not a guarantee to earning a reward. Congratulations, you earned 100 points today. Keep up the good work, or sorry, you didn't earn anything. So that kind of Skinnerian conditioning and that intermittent reinforcement we felt was something quite powerful. And so we wanted to deploy it, but not do it in a lottery-based system, but rather do it in a smart way where we could actually algorithmically increase your adherence while lowering the overall cost of paying you to comply with your medication adherence. So that's what we call dynamic intermittent reinforcement. And the unfair advantage that we had over the competition was the data that we could collect and the ability to learn from each patient and tailor the algorithm specifically to you. Okay, so just a basic example of what is the problem, articulated both from the patient's perspective as well as in economic terms. What is the solution to the problem, appropriately explained for the audience at hand? And then what is the unfair advantage that that solution has relative to the competition? Okay, so once you've articulated, that's all step one. Once you've articulated that internally to yourself, then you take step one, and step two is taking step one to the tech transfer office, communicating what you've articulated to yourself to your partners at Ventures. And I'm just going to assume that Ventures is your tech transfer office for the purpose of this discussion. It could be a different tech transfer office, but I'm going to assume it's Ventures. And the easiest way to take your invention there is to email Ala at the email address provided. There's a website link I'm providing. There's a great guide to all the processes associated with having to do this and all the forms you can download, et cetera, through these links that I provided. It's important to do this because you have to do it, because the technology you're developing is actually not your technology in an exclusive sense. It actually belongs to the university. So you actually have to take it to the university and together and collaboratively work with the university in order to think of the best ways to commercialize the technology and really scale it up to help as many people as possible. So the next step then, step three, is to work with Ventures to determine if your tech is patentable and has freedom to operate. Now, there's some great resources to better understanding patentability and FTO, intellectual properties. One of the previous talks in this series is specifically on that by folks that are a lot more knowledgeable than I am. But at a very high level, the patentability is simply determining if your discovery or your technology is new, non-obvious, and useful, whereas freedom to operate is the flip side to that coin, determining whether your invention is infringing on existing claims from in the prior art. In other words, previous patents that have been issued that have claims that potentially cover some part or all of the technology you're trying to develop. So it's important to develop to an understanding of whether the technology you have is protectable. And that comes in two flavors, both patentability as well as freedom to operate. The source that I use often is lens.org. There's many sources, including uspto.gov. Lens is just a great free resource that gives you access to a whole array of US and international patent databases, and is pretty easy to use from just a user interface perspective. Now, it's at this stage, prior to filing IP, that, and with the help of whether it's your entrepreneurs in residence colleagues, your tech transfer office, or your patent attorneys, to determine whether it's necessary to perform additional experiments to discover results that, quote unquote, teach away from prior art, because that will enable you to develop stronger IP. Okay, so, and we can talk more about that strategy, which is not often, used. So, the typical way to do this is, the academic researcher comes up with some discovery, they go to the tech transfer office, the lawyers get involved, and then there's a lot of work that's put in with the experiments at hand, as opposed to real forward-thinking ways to develop and really look for results that teach away from prior art. In other words, are unexpected, because that will strengthen the IP and sort of reinforce yourself to build a case against the inevitable sort of queries that the patent office is going to send your way to, and that you can defend yourself against. Okay, so once you start working with Ventures and you file the IP, the next step is to determine whether the strategy for commercializing technology is going to be out licensing the IP to an established pharmaceutical company or other larger company, versus starting a company around your IP. And that could happen through licensing it to an existing startup, and as the foundational technology for that startup, or developing it as a spin-off from the university itself. Once that determination is made, and again, moving forward, I'm going to assume that we decided to move forward in starting a fresh company, just from scratch. So once the decision has been made to move forward to starting a company, then the university has a very rich entrepreneurial ecosystem that you can tap into to move forward to starting that company. And I've listed a handful of the different areas through which I've become familiar over the last month or so. I guarantee you it's probably not an exhaustive list of the resources that are available to you. Some of these are actually state resources or county resources, as well as university resources. But again, I think the best way to familiarize yourself with many of these is to go to this link here, startupguide.umd.edu. All and colleagues have really put together a terrific guide there. And of course, just, call up tech transfer office, really get these discussions going earlier rather than later. It can be confusing, I would suspect, because of just the vast richness of the ecosystem that's available to you. But you've got folks and guides and Sherpas along the way that can sort of, give you a sense of what makes sense for you. Okay. Now, we're going to move forward. Are there any questions so far? Because what I'd like to do is spend the majority of the conversation today on what happens after, right? So that I think there's a terrific resources available to get you to this point of understanding, of forming the company formation through the university. Maybe relatively less perspective on what follows after, specifically for biotech. So before I move on, are there any questions along those lines? Okay.
I don't see any questions posted, but please do feel free to submit them, or you can also raise your digital hand and I can unmute your mic. I don't see any at the moment.
Okay, let's move on. That's great. Okay, so the template for moving forward from company formation onwards in a biotech really comes down to having a real good sense and appreciation of the investigational new drug application. The different parts of it are shown here. They come in 5 modules, and we'll talk about each of these in turn without making it a real exhaustive regulatory discussion. I put this up here because I believe that biotech is such a regulated industry that any entrepreneur that's looking to start and grow their biotech business, I think has to have some intuitive feel as to what the IND is, because it speaks to what the FDA's expectations are, and it gives you a template of to follow as you build your company and develop your technology. Okay, so module 1 is administrative. Module 2 are the summaries, both non-clinical and clinical. The non-clinical components come down to really 3 main categories: pharmacology, which is all the efficacy evidence that you accumulate that suggests that your drug is working; pharmacokinetics, which is the absorption, distribution, elimination, metabolism of your drug in vivo, whether it's the patient or in animal models; and then toxicology, how safe is your drug in particular in preclinical models. So there's different sections, and we can talk about it in more detail, but that's sort of, it's important to, in module 2, to present your arguments as to why you believe the drug works, what the kinetics of the drug are expected to be in humans, and what the safety of your drug is based on your animal toxicology work. Clinical summaries are also included here. Often in a first-in-human drug, obviously you're not going to have a lot to talk about with respect to your drug, but you can certainly talk about the field in general and other similar drugs that have been in the clinic, to give the reviewers, the FDA, a really good sense of what would be expected when this drug gets into the clinic. Module 3 is how you make your drug, chemistry, manufacturing, controls. 3.2s is a drug substance. 3.2p is drug product. That includes the formulation for your drug substance, the stability at different conditions and different temperatures, and also packaging. If you're building out a facility, 3.2a needs to be included. So that's module 3. Module 4 are all the study reports for all the studies that you've generated internally, whether it's analytical studies or non-clinical studies, et cetera. Those go into module 4, as well as any interesting papers that you want to include that support the story. And finally, module 5 is the clinical section. It includes the clinical protocol, informed consent form, and a few other forms. So I encourage you all to sort of take a look at this. Lots of information is available online. The link I put is directly from FDA, so you can't go wrong with that. But having a good sense of what the FDA expects along the drug development process, I think will make you a better entrepreneur. And the preclinical line, which is what we're going to talk about next, goes from discovery to the FDA sending you a study may proceed letter and allowing you to move forward. So that IND allowance marks the end of the preclinical development path for an asset and the beginning of the clinical development path. Okay, so again, a few examples through this the different company, Topokine Therapeutics, where we were developing prostaglandin F2 alpha analogs to reduce fat topically. So here's back what we were thinking back in 2011, 2012 in terms of the development path to get it from discovery to the IND and phase 1 clinical studies. And we broke this into 2 different sort of ways of thinking. The red is all the stuff that happened prior to initiating our GLP toxicology studies. GLP stands for good laboratory practice, by the way. And the GLP, and then, the blue is everything that happens once you initiate the GLP tox. So we, up until this moment, we had developed enough evidence internally to be convinced that our drug could reduce fat. Okay, and I'm going to show you some evidence associated with that. So the red here meant that now the work that was left is to optimize the formulation for this drug called XAF5. It's basically an ointment of prostaglandin F2 alpha called latanoprost that's used for glaucoma to reduce pressures of the eye. And we saw that applying it directly on skin reduced fat topically without surgery. So we wanted to optimize that formulation. So we allocated some costs associated with that, and we figured that would take a few months. We needed to develop the methods to be able to measure this drug in the body of our preclinical models as well as in the clinic once we got there. So we wanted to take a few months and allocated some dollars to doing that. And we wanted to build in some money to go and ask questions to the FDA in the form of what's called a pre-IND meeting that gave us a real good appreciation of what the FDA was thinking before actually filing the IND after completing all the work in blue. So, and then once we did that, we wanted to leave some room for making the product, determining what the stability was, using the product that we made in GLP toxicology studies, determining what the pharmacokinetics was, and then finally preparing and submitting the IND. The IP stands for investigational product, not intellectual property in this case. Okay. So let's just go through each of these steps here. So the pharmacology that we developed for the IND is listed here. We saw that our product was capable of reducing fat in cell culture, capable of reducing fat in rodent models and mini pigs. You can see the before and after pictures. We saw that it reduced fat in African green monkeys when applied topically on the belly of these animals. We ran a toxicology study, and this is a report. I just copied and pasted directly from the report provided by our contract research organization that did this toxicology study where we applied the drug on a regular basis and determined that what the no observed adverse events level was, the NOEL. So basically that the objective here in a tox study, a GLP toxicology study, is to determine what determine the dose right before you start seeing adverse effects, because that's the dose you're going to take divided by 10 and use to make a justification as to what your starting dose in the clinical trials is going to be. Okay. And then once you determine that, you can write up, again, this is all preclinical, this is going into the IND, what your phase 1 clinical development plan is going to be. You can use it to determine what your initial starting dose is going to be in a single administration. You can determine what it's going to be in multiple administrations, in multiple ascending dose study, and really come up with an appropriate rationale and justification as to what your phase 1 clinical development should be based on all the data that you've collected, both in your pharmacology studies, your pharmacokinetic studies, and then finally your toxicology studies. Okay. So in this example, you can see that our clinical development plan for this phase 1 study was going to administer the drug at different doses for, up to 6 weeks at one side of the belly as opposed to the other, and, look at plasma pharmacokinetics, CT scans, et cetera. And so once we did that, we put it together, we submitted an IND, and a lot of back and forth happens in the 30 days that the FDA has to respond to your application. And during those 30 days, depending on the division, you often get a lot of different queries from the clinical reviewer, from the non-clinical reviewer, from the CMC reviewer. They really want to understand in detail that and so there's a lot of back and forth. They ask questions and you respond. And with a little bit of luck and a lot of preparation, you might get a letter like this, which the key phrase is that this part here is the FDA has concluded that your study may proceed. Okay. So that's great. So you get to the point you've finished preclinical development, what comes next? Now, before I move on, are there any questions so far on the preclinical side?
Don't see any questions, but the question I would like to ask is it different time-wise and also development-wise, steps-wise, if somebody is developing something that goes inside the body rather than topical?
Right. Yeah. So this is where the short answer is yes, but it's not entirely predictable. What your timeline is not entirely predictable based on the route of administration. So whether it's an eye drop or a topical or intravenous administration or local, intracerebral administration, intrathecal administration, there's a whole array of different routes of administration. And the development, whether it's preclinical or clinical, is entirely dependent on the interplay between the risk and benefit associated with that particular drug. There are some mechanisms that allow you to go very quickly if there's already a safety and efficacy package for that drug, but for a different route of administration. So, like, for example, with Topokine, the example that I was giving you, it was a drug that was actually administered intraocularly, or topically, but as a drop in the eye, and it was a commercial product called Xalatan. It's still used; it's very popular. It's used to reduce pressure of the eye, and as a result, Pfizer had done a ton of work to determine the safety and efficacy. And so we had, by virtue of the fact that FDA had access to all of that, we could pursue a mechanism called 505(b)(2) that allowed us to just, go through the preclinical development very quickly. If it was a brand new drug that we were developing on the skin, or intravenously for that matter, it would have taken a lot longer. It would have been more expensive. So yeah, it all depends on what the body of evidence is moving forward in your clinical development, in your preclinical development. Okay. So
We have
the
another
sorry, go ahead.
question. Sorry. So another question here. Let's see. Would you say that the FDA is communicative? If so, not how do they go about communicating and aligning themselves administratively with your company? For example, do you get a case manager?
Yeah, that's a great question. So, so the process is, once you've made a determination that you're going to file an IND application, you register you let the FDA know of your intent to do so. And then you get an email back from them that assigns you a specific staff member, right? Like you said, a case manager, if you will. That individual is the middle person between you and your company and the three different review groups: the clinical, the non-clinical, and the CMC review groups. Often all communications go to that person, from you to them, and then from them to you. They all cross that same individual person for that particular IND. So the 30 days that you have, you're getting a lot of emails from that person. Okay. Never or often not from, any given reviewer. I say often and not never, because I do remember one time, for a different company, Cartesian, that we'll talk about, that in the early days, I was in my living room and I got a phone call during the 30-day review period from the actual CMC reviewer. And we just had a very friendly chat because he was confused about some specific thing. So it really depends on the division. And this is through an Office of Advanced Therapies for Cartesian, for cell and gene therapies. It depends on the process that each division has internally, and it depends on the personalities of the different reviewers. But the general principle is that you're assigned an administrative individual who is going to be the middle person between you and the different reviewers. It's a great question. Okay, great.
Thank you.
So once you're in clinical development, there's different phases. These are the classic phases, phase 1, phase 2, and phase 3. But of course, there's a lot of interplay between these different phases, and we'll go through some of the different considerations. So classically, phase 1 is safety, pharmacokinetics, and correlates of efficacy, where the goal being to establish the maximum tolerated dose, which can often, but not always, be the phase 2 starting dose. Okay. So your objective in phase 1 is to dose escalate to find what the maximum tolerated dose, and there's definitions around what that means, for that particular population of individuals. Sometimes, often, these studies are done on healthy volunteers. In other indications where the risk benefit may not justify treating healthy volunteers, for example, a lot of cell and gene therapies, CAR Ts, et cetera, you start right away with, and in cancer, you start right away with treating patients, even in phase 1. But the objective remains the same. What is the maximum tolerated dose, and so that you can start to discover what the phase 2 starting dose might be. Because once you get into phase 2, while safety certainly continues to be paramount, efficacy becomes important. And a huge milestone for any biotech company is to establish clinical proof of concept, to demonstrate efficacy in the population relevant to the indication relevant to that drug, right? And with the goal of phase 2 being to establish a phase 3 commercial dose and dose schedule, and to lock in a commercial manufacturing process, because then the only thing standing in your way in a marketed product is a phase 3 study, one or more phase 3 studies, with the goal being to establish efficacy definitively in one or more randomized controlled trials. Okay. So now there's designations: phase 1, 2, phase 2, 3, phase 2B, phase 2A. They're, in essence, I wouldn't worry so much about those designations. These days, for example, in oncology, if it has any sort of efficacy component, a phase 1 study might be called a phase 1, 2A. But in essence, these are the general categories. There's no regulatory meaning associated with these A or B designations. That may be helpful to your investor audiences and others, but ultimately these are the different phases. They're classic, and it doesn't mean that you have to complete all 3 phases to get approved. There's a lot of cancer drugs that get approved after phase 2. And an accelerated approval. There may be some additional what are called phase 4 studies that are done to support the continued safety of a drug after accelerated approval. There's different regulatory designations to allow you to be able to go through things faster. If it's a very promising drug, a breakthrough designation, or RMAT designation in cell and gene therapy, regenerative medicine advanced therapy designation. But without complicating things, and for purposes of today, these are the different phases that the agency is expecting. So here's a clinical development plan, again, for the same company, Topokine, that we came up with when we went into the clinic. So in red, phase 1, phase 2, phase 3 are outlined. It doesn't mean that your non-clinical work stops once you get into the clinic. You continue to support your clinical development with work that you put in non-clinically. So if you want to do a 9-month treatment, you need to have support from non-clinical GLP toxicology study to be able to treat your patients for 9 months. But sometimes it doesn't make sense for you to go and, treat animals for 9 months, wait, and then get into the clinic. So you can sort of time these in a way that allow you to get into the clinic relatively quickly, and then start longer-term toxicology studies and preclinical models that give you support at risk for you to be able to treat patients for longer periods of time. So just gives you a sense of what these things look like. The STAR is an end of phase 2 meeting. So you have opportunities along the way of clinical development to go and get feedback from the FDA. And as I mentioned, there's different regulatory mechanisms that allow you to be able to do that more efficiently if your drug is particularly promising. Okay. So again, for topokine, so here's that abdominal fat study. So this is a phase 1 study, but we also wanted to collect correlates of efficacy during this phase 1 study. So we did scans of these individuals to look at levels of fat along the way, and saw that at a high dose of applying this gel, we saw a reduction in subcutaneous fat in these individuals. And that gave us the confidence to move forward and propose a phase 2 clinical design, where we propose to take this now ointment and apply it on a daily basis for a couple of months to see if we could reduce the fat in underneath the eyelid of these individuals that had what's called steatoblepharon, or excess lower eyelid fat. We developed and validated a scale using a professional artist, and along the way gave placebo versus XAF5 ointment and determined that we could reduce subcutaneous fat in these individuals. You can see the before and after photos after 70 days of treatment. In a couple of representative patients, you can see this fat here reduced as a result of applying this ointment. And that gave us and then we looked at it different ways and methods, and we saw that there was a statistically significant reduction in subcutaneous fat, measured both from the clinician's perspective as well as the patient's perspective. The FDA really likes to have these patient-reported outcomes to sort of reinforce the efficacy and the relevance of the therapeutic to the patient. So once we put all that together, we had our positive clinical proof of concept, and then had a meeting with the FDA and reached concurrence with the FDA to move forward with a phase 2b/3 study design. So this is a larger study. Instead of 90 patients, this is 300 patients. But the general idea remains the same. We're treating these patients a little longer. We're introducing yet another dose, but this now could be used to support registration and a biological or new drug application to get licensing approval to market your drug. And so we designed a study to be able to show a difference relative to placebo to be able to do that. Okay. So that's in essence the very short road or very short explanation of the getting from discovery all the way through phase 3 studies. I want to take a few moments and shift gears and talk about some topics that are relevant all along the way, with the first being, what kind of company do you want to build? Is it going to be a virtual company where you're outsourcing most of your operations, if not all your operations, to different contract research organizations or manufacturing organizations? Or is it going to be a brick and mortar operation where you build, a manufacturing facility, for example, and bring in-house clinical operations and quality team and R&D? Or is it going to be something in the middle? The pros of a virtual operation is that you can keep the costs very low up front, but the cons is that you're entirely dependent on vendor partners, and that can become quite expensive in the long run, both in terms of mistakes that your partners can make, as well as the costs associated with the additional payments that you would have to make in order to, support their business model. They're going to charge you more than what you could do in-house, of course, once you've acquired the costs to be able to do it in-house, the equipment and the facilities. You can do things a lot more cheaply relative to what a given outsourcing model would be. For a brick and mortar model, a more specialized team with in-house core capabilities has a lot of pros. You can move very quickly once you've established that model. It's very collaborative, highly innovative, efficient operations, rapid innovation. But the cons is that there is a significant upfront capital investment that you have to make, as well as the maintenance costs of that investment along the way. So, often, we see hybrid companies where there's certain select core competencies you bring in-house, for example, the R&D, with other competencies being outsourced, for example, manufacturing. So another way to think about it is that as an entrepreneur, you're going to be asked not to be an expert in all these different areas, but rather to be able to ask questions of your partners in these areas, whether they're internal resources or external CROs, and to be able to align and manage and balance these different resources and these different specialties, right? So again, you're not necessarily an expert in any and all of them. You're probably going to have your strength in one or more of them. Often, it could be an R&D expertise, but along the way, you're going to want to get comfortable with a whole array of things and step outside your comfort zone to learn and be really interested in things that you otherwise would not necessarily find interesting. So I'm going to give you just two quick examples of how we did this. We've done it both a virtual way as well as a brick and mortar way. The company we were talking about, Topokine, this is fat reduction company. We started that with just the two of us. Mike Singer is my longtime friend and partner of 20 years. And that is a photo of the two of us from, I think, 20 plus years ago. And when we started that company, it was just the two of us. And when we sold it, it was actually just the two of us still. We had outsourced everything. We just worked out of our homes. We didn't even have an office. We outsourced the preclinical R&D. We outsourced the toxicology studies, the analytical studies, as well as the clinical operations, the manufacturing, everything, absolutely everything. That's very different than the company we started a week after the sale of Topokine, which is Cartesian, is a cell and gene therapy company, a CAR T company for autoimmunity, where we brought in a third co-founder, Metin, who's currently our chief technology officer, and wanted to apply our virtual model. In fact, began in 2016 trying to outsource preclinical R&D. Realized that wasn't going to work. So we said, okay, well, maybe a hybrid model will work where we'll just get some lab space, do some process development, and as well as some R&D, but really we'll outsource the contract manufacturer. We'll find a contract manufacturer and outsource the actual GMP manufacturing. And we started doing that. And after our first IND was allowed, we actually even treated a handful of half a dozen patients with material that a third-party contract manufacturer made for us. But we realized it was painful and the product wasn't good, and so it wasn't good for patients. The kind of back and forth with the contract manufacturer didn't make sense. It was actually more expensive because they were charging for all these things that we didn't agree with. So we made a strategic decision to bring manufacturing in-house. We found some lab space, just state-of-the-art space just across the street from NIST in Gaithersburg, built our manufacturing facility from the ground up, and as a result had to bring in hire individuals that could run the manufacturing operations. We built a whole quality department. And then we thought, well, why stop there? Let's do clinical operations internally. We used to outsource that and to a CRO, but then, we said we could do that too. And we brought in clinical operations. So what grew out of a real desire to build out Cartesian virtually transformed over several years into a brick and mortar operations where, we now actually have these core competencies in-house where everybody's under the same roof. And that we've certainly realized the benefits of being able to have control over our manufacturing and really the processes and the timelines, et cetera, in ways that I think we would have had a very difficult time doing. And that's particularly important in cell and gene therapy as opposed to more established disciplines like, small molecule development or even biologics development like antibody development. So the entrepreneur really needs to sort of, I think, feel comfortable get comfortable outside your comfort zone and be able to be flexible, to be able to shift these different operational models relatively quickly when you realize one model isn't working, to be able to shift quickly to a different model that hopefully will work. Okay. A little bit about financing. Now, we're not going to have time to get into this as a whole talk in and of itself. I did want to just, I do get asked, like, how much does it cost for preclinical, early clinical, late clinical? Well, from the previous couple of slides, you can surmise that these numbers are going to vary wildly depending on the kind of operations that you intend to pursue. But I put some, very large ranges to just at least give you a sense of what the kind of dollars that you may expect are. The sources of capital are, I think of it in two different categories: conventional and alternative sources of capital, with the conventional being the sort of the typical things you think about when you think biotech capital, venture capital, strategic investments from big pharma. That certainly is something. Crossover investors, which are investors that tend to do most of their investing in public companies, 90+% often, but do have some allocation for private companies, and they like to invest in those companies that they think will potentially go public relatively quickly within 18 to 24 months. Private equity, and then public markets in particular, where you can have access to very large pools of capital. The pros of conventional capital are lots of capital, brand power, and the network. Compare that to alternative sources of capital. I think of this in three different subcategories: investors, grants, and strategic licensees. Investors, friends and family, angels, family offices. Grants, completely non-dilutive in universities, foundations, county, state, or national governments. And of course, strategic licensees who are interested in licensing some of your technology or sub-licensing it, and as a result can actually give you working capital that you can deploy to other programs. So the pros here for alternative capital, there's less dilution and more control. There's no right way here, but those are the two main sort of sources, categories of capital. Capital structures are also varied. Grants versus contracts versus convertible securities. Those come in two flavors: notes versus safes, which are really not like notes or debt, but rather promises to future equity. Relatively simple arrangements for younger companies in particular that really don't, the investor doesn't necessarily have a good sense of how to price the company or the value of the company. Once you get to a later stage, often, you do price rounds, whether it's issuing common stock or preferred stock. Lots of different ways to do this. Again, these are sort of, entire talks in and of themselves. Valuations, we get asked, like, how do you value your company? Well, there's different value inflection points along the way. I've highlighted both preclinically, clinically, and commercially. I've highlighted, the ones that I think personally are big value inflection points. You get to an IND, that's great. You're now a clinical company. Once you're a clinical company, you've got POC, proof of concept. That's a huge value inflection point. And finally, marketing approval. There's other smaller value inflections along the way. And as you would expect, to go from preclinical to phase 1 to phase 2, the valuation of your company should increase commensurate to the development that you're pursuing. And finally, last but not least, is exit. You can think about, and this is where you have some liquidity. So before you're issuing shares or the promise of issuing shares to your investors, you've raised capital. What is, ultimately your investors are looking for an exit. You can think of an exit from the perspective of stage of development, preclinical versus early, mid, or late stage clinical versus commercial, or the two key types of exits are M&A, an acquisition, versus going public. We've done both of these. So the Topokine company, the medical aesthetic company, was acquired. So that was a typical M&A that had an upfront payment and back-end milestone payments, depending on whether the acquirer was able to hit certain regulatory and commercial milestones, versus taking Cartesian public through a reverse merger. We had, in essence, a public company that we merged with, and as a result, we acquired their capital as well as their management. Own the majority of, so we own the majority of that, and now public company. But as a result of that merger, we're able to capitalize our company. Going into it, we had relatively little capital. After the merger, we had upwards of nearly $120 million in capital. And that's important because things get expensive as you get closer to commercial development. And so it's important to think about exits from the perspective of how do you capitalize your company moving forward. So accessing the public markets and the ability for your investors and your employees to be able to sell your shares, it's an important part of the journey in raising capital. I'm just going to end with this slide. So this is a high risk business, folks, right? So only 1 in 15 cancer drugs that reach clinical development, get to phase 1, actually reach the cross the finish line and become a marketed product. That number is about 1 in 8 for non-cancer therapies. And companies can fail for many reasons. I've listed a handful of these here. So what balances that high risk is high reward, both intrinsic, because it feels really good to contribute to treating or curing a disease, as well as the extrinsic, the financial independence for you and your loved ones. This is not something to be embarrassed or ashamed about. I mean, I like money. I assume you like money too. And to borrow from one of my favorite movies, Idiocracy, I can't believe you like money too. It's like, it's fine. Everybody likes money. And so this is an important contributor to ensuring that this model works, because if it's high risk, it really the venture needs to offer a disproportionately asymmetrically high amount of reward if things do tend to work out. Okay. So now with that, I'm going to stop. I know we're at the time point, but I want to see if there's any last minute questions. And again, we can continue this conversation either through Ala or Ala. Feel free to share my email with anybody who asks as well. So with that, let me stop and see if you have any questions.
Thank you so much. This has been really fantastic, and I know an overview, and I know it's a huge topic. So thank you for distilling it into such a short time. But we do have a few questions. So here's one. What would you say were your first important strategic decisions that you made in your startups? And who were your first hires and why did you need them?
Right. Yeah. So it depends. So let's use Cartesian for an example. So we already talked about some of the strategic decisions in terms of the virtual versus hybrid versus the build, right? That sort of the brick and mortar build. And as we shifted the model, the hiring decision certainly reflected that, right? So when we made a decision to build out an internal manufacturing facility, we had to bring in a head of manufacturing and a head of quality. When we made a further decision to bring in internally our clinical operations, the execution of the clinical studies, we wanted to bring a seasoned head of clinical operations into the team. So it really depends on there's no one right answer. Again, it kind of depends on the model that you're pursuing and what you want to accomplish along the way. If you'll remember that diagram with the entrepreneur in the middle and all these different sources, every single one of those smaller circles that circle the entrepreneur can be outsourced, right? But it doesn't necessarily mean that they have to be. And I would argue that if you're having for those core competencies that you feel are absolutely critical, it makes sense to bring them internally because things will just happen faster and more smoothly relative to if you're outsourcing to a vendor who has 7 other clients like you do, who doesn't report directly to you, et cetera.
Thank you. Here's one more question. Aside from the firm's inherent technical challenges and funding pursuits, a classic question with respect to the process outlined today, what keeps the entrepreneur or the burgeoning firm's leadership up at night?
Right. So aside from the technical challenges and the funding, right. Yeah. So I would say that there's 100 decisions that you have to make on a daily basis. And the total sum of all those decisions is what kept me up at night across these three companies. In other words, what am I missing? What am I not thinking about that's going to blow up in my face? There's going to have to be a fire that I have to put out. See, it's I think kind of important. So this is kind of interesting. This is the entire weight of your investors, the pressures that you put on yourself are on your shoulders as the entrepreneur, right? It's just not it's not a lifestyle job. It's not a job. It's not even a career. It's a mission, right? You're on a mission to build and develop a technology you believe is going to be potentially curative for potentially awful disease, right? Um, so, the buck stops with you in all respects. And as such, you have to be sort of on, 24/7 moving forward. And that could be quite stressful if you let it get out of hand. Now there's a, it's a bit of an illusion, of course, because all you can really do is the next thing. But ultimately, especially as a sort of in my earlier days as a young entrepreneur, sort of let that kind of get to you and things can get kind of stressful. I would just encourage you to sort of take a step back and realize that all you really can do is the next thing. You can't think of 100 things simultaneously. And to just, take time slowly and deliberately, fully cognizant of the fact that you're going to make 100 decisions a day. And as long as you get 80 of them right, you're doing okay. And, as long as you surround yourself with folks that are supportive and are able to sort of give good advice along the way, and as long as you're able to triangulate the different opinions that are coming your way by asking really good questions, and again, becoming interested and familiarizing yourself with that ecosystem that you build around yourself, you're going to do okay.
Thank you. Let's see. I think that's all for the questions. And let me see. Again, thank you so much for sharing this. This has been a fantastic workshop for everyone. I will share the link to the video when we upload it to the YouTube channel. And for those who haven't had a chance to attend live, I know that there are many of you watching it on YouTube. Please feel free to contact either Murat or me with questions and I can forward them. Again, thank you so much, Murat. It has been fantastic.
Thank you, Alla. Thanks, everyone, for your interest.
Yes, and thank you to everyone who attended and asked great questions.